I'm François Delmotte. I'm working at INRA Bordeaux at ISVV, and here I will present you. I've tried to present you some of the results we had the last 8 years in our lab, and of course during the course of the project, especially at the end of my talk. So the aim here is to give you some key to the idea of sustainable, durable grapevine resistance, with a case study, a special focus on downy mildew. I think that decreasing significantly fungicide today involve a necessary breakthrough, an innovation in the training system of grapevine in Europe, and actually, to my idea, the only innovative and promising innovation would be to use natural grapevine resistance against pathogen to reduce significantly fungicide. So as Bruce already told, so this slide is just to illustrate that we have a lot of sources of resistance in Vitis species compartment. And especially in North America, where the pathogen grapevine downy mildew is coming from. So today, new grapevine varieties showing low to high level of resistance, I mean from tolerance to highly resistance to powdery and downy mildew, are registered in some countries and in Europe, in France soon. And well, contrary to all the interspecific hybrid, these newly bred varieties have good cultural and enological features that allow to grow them in vineyards. So the idea now is to move from a cropping system based on pathogen control, based on plant manipulation, as you know, biological control, but mainly about chemical control, toward a new system where we will have a more balanced distribution of the role of each of them, integrating plant disease resistance in the system. So we have tried to assess such new training system based on a resistant variety, an INRA resistant varieties in Bordeaux, where we have planted about 0.6 hectares of a genotype, which is fully resistant to powdery mildew and tolerant to downy mildew. And this long-term experimental approach has allowed to assess a lot of performance of the system, such as agronomical performance, economical performance. And here I present only the features on the treatment frequency index of these systems. So it's easy to see that the reference is Bordeaux. In Bordeaux vineyard is this one, where we have about in Bordeaux in 2015 we had about 14 different treatments. An organic system we had also in this experiment reached a decrease of 20% of treatment, but here you can see that the resistance system really behave differently. It's a complete breakthrough because we have that has allowed 90% reduction in fungicide. So this is a big hope for viticulture. However, there is the other side of the coin, which is durability of the genes. Because we need to make sure that the resistance employed is not only effective against the pathogen, but also that this resistance persists in time despite the constant evolution of pathogens. So Bruce Reich told us about the durability of resistance of powdery mildew. And this is effectively something that happens in our vineyards for downy mildew in Europe. The durability of resistance is especially important for perennial crop like grapevine, which are meant to stay in the field for 20 years or more. So, and we know that from many other pathosystems that have been studied that plant air genes are not everlasting, never. So this is just a slide to show what kind of, usually we distinguish two kind of resistance. The total resistance, Here, the first picture, where we have an evolution of the pathogen from total resistance to full susceptibility of the plant. And in the case of partial resistance, it's not a breakdown of resistance, but it's an erosion of the resistance of the plant. So there are some clues that we can make about the durability of resistance. The first one is maybe the nature of the resistant genes that are involved in the interaction. I just told about total versus partial resistance. But also the evolutionary constraints that are related to the modification of the corresponding gene in the pathogen that allow it to break down the resistance or to erode it, the so-called cost of virulence. But also there are some important insight to get if we want to translate it in terms of durability of resistance, is the evolutionary potential of the pathogen, which is related to its genetic diversity, its reproductive system, etc. So that's what we have tried to do for grapevine downy mildew. So Plasmopara viticola is a native species from North America, which has been introduced in the late 18th century. But it's in its native range, Plasmopara viticola attacks a large range of vitis species. So we get back to this native range. We sample the pathogen with the help of a lot of colleagues in the United States, especially Lance Cadel-Davidson, David Gadori for Cornell University. And we get a new picture of the pathogen, because actually Plasmopara viticola in North America is not just one single species, but a complex of several cryptic subspecies that are more or less all specialized on wild and cultivated grapes. So I try to make it short here. So you see, yeah, there are at least now 5 taxa that have been identified. They have been found on several wild grape. But maybe the most important to say today is that Vitis vinifera, so the European cultivated grape, is in this picture related to the first clade here, which I've called vinifera, and a second one here, which is called estivalis. So at least 2 of these species are able to attack Vitis vinifera. So the question for us was what was introduced at the end of the 20th century in Europe, and the analysis is clear. Here you see the diversity at one gene, the tubulin gene, in the American populations, with many haplotypes. I think it's 80 haplotypes. And for European isolates, we have screened about 1,000 isolates in Europe. We only have 4 haplotypes represented in the tree, and they are all coming from one species, which is Forma specialis estivalis. So a very strong bottleneck at the introduction into Europe. In Europe, the genetic structure, as we said yesterday, is very low genetic differentiation, only a slight but significant continental isolation by distance, as you can see with this figure here. Nothing very spectacular, nonetheless. And when you look at the distribution of the population across the world, you can see that introduction occurred from North America to Europe, and then Europe spread the pathogen across all, let's say, new vineyards like South American vineyards, South African vineyards, and Australia. That is the picture we have now. Actually, the risk is in Europe is related to a reintroduction of the pathogens, since we only have one of these species that is attacking the Vitis vinifera, the cultivated grape. But also now we know that there is an efficient sexual reproduction of the pathogen. There are also large population with large effective size and important gene flow at a large geographical scale, which together fits to say that this pathogen may have a high evolutionary potential. And indeed, Plasmopara viticola is evolving in response to plant resistance, as demonstrated by a collaboration between the University of Udine and INRA about the resistance of Bianca, since these teams identify one strain of Plasmopara viticola that is able to break down the RPV3 resistance. To go further in this evaluation of the evolution of the pathogen in front of partial resistance of grape, we have tried to design a new protocol, let's say, by sampling isolates on both Vitis vinifera population and also on the resistant varieties that have been legally deployed in Europe, but still in Germany and Switzerland you can find them. We have re-inoculated the host and the pathogen in the lab and measure quantitative traits of the pathogen to see if there is something occurring in Europe too. So here is the sampling design. So as you see, we sampled where the resistance variety are deployed in Europe, not only in experimental vineyards, but also in some vineyards in production, like in Germany, which have been planted for more than 10 years for some of them. And we cross-inoculate then the pathogen with the plant. So above the pathogen here, that came from 11 different resistance varieties and were re-inoculated on Bronner, Prior, and Regent, and also for the control Cabernet Sauvignon. So to make it short, here is the kind of experiment we had during the course of Innovine, huge cross-inoculation with a lot of pathogens and a lot of plants, and where we assessed sporangial production, sporangia size, latent period of the pathogen, sporulation dynamics, zoospore numbers, etc. But here only present a very summarized result of this experiment. So you have here two kinds of population, as I said. The naive population that was sampled on Vitis vinifera, susceptible variety, and the adapted, let's say, potentially adapted population that was collected on resistant varieties. So in light green, you can see that grapevine resistance is really efficient to control the pathogen in the lab. Bronner is bringing an 80% of control of the sporulation compared to a susceptible grapevine variety. This is QTL is RPV10. And for Prior and Regents, it's between 60 to 70% of the sporulation. So a really efficient control of the pathogen by plant resistance. But when you look at adapted population, dark green in this bar plot, you see that this population is less well efficiently controlled by the resistance than the light green, including for Bronner, Prior, and Regent, for the three of them, with a reduction about 25% in the case of Regent and Prior, the RPV3 QTL. Also, when we look at the source where we have sampled the source of resistance where the isolates came from, we could tell that there was no specific adaptation of the pathogen to its variety. The isolates that were collected on Bronner were not more aggressive on Bronner than on Prior or Regent, which means that the pathogen is maybe not adapting to the specific QTL, but more generally to partial resistance, being more aggressive in general on these resistant varieties. Another interesting result is that these adapted isolates are not genetically differentiated from the others. They are completely in the genetic background of the population, of the Plasmopara population. It's not a clone that invaded the the field or the vineyard is a lot of genotypes that are able to be more aggressive on these plants. And to go further, we are now trying to look at the genes that are responsible, that may be responsible within the genome of Plasmopara viticola of this erosion of resistance. So we have sequenced, we have just released the Plasmopara viticola genome. We are now resequencing isolates in order to see if we can detect in the genome the locus that are involved in this erosion of resistance of RPV3. So what can we do now? Now we have said and we have seen that the monitoring is, that the pathogen population are able to evolve. What can we do? So my first advice would be to try to have some more theoretical approach on that, questions of the durability of resistance. For instance, trying to have some modeling approach in order to optimize the deployment of this resistance in the field. There are several questions we can try to answer. The first one would be on the interaction between the pathogen and the plant. What are the pathogen traits that more impact the erosion of resistance? And if we have to select for a more durable resistance, it would be interesting to know which of the pathogen traits are more involved in this aggressivity against the plant. So we can do that by a modeling approach using the data we have produced in experimental conditions. Another important question is how to manage these genes when they are deployed. You know that we have talked about stacking the gene in the plant, but it's a long work, and it's also at the expense of many other traits. You cannot do everything in one cross. So maybe we have to explore more situations where, for instance, we can deploy the genes in mixture or a mosaic of genes in a landscape. And that's what we are going to explore with this model developed by Frédéric Fabre at INRA Bordeaux. Another important issue is the cropping system. The idea is here not only to produce systems that rely on natural resistance, and it's something we would like to do and just not treat the plant, but I think that we have really to combine different practices, including agronomic ones, prophylactic ones, fungicides, and to apply the more selection pressure we can on the pathogen. So the plant is one of these selection pressure, but we really advise to try to use also some fungicide at key development stage of the plant in order to limit the adaptation of the pathogen. Also to complete the efficacy of plant resistance in order to secure the production in bad climatic or highly conducive conditions for the pathogen. And also to control the pathogens that are not targeted by resistance, such as black rot or anthracnose, as we discussed yesterday. Because these pathogens in these low input systems are really emerging, and they are able to destroy the harvest if they are not controlled. So here, this is my last slide, just to remind that the idea is that plant resistance is obviously a promising solution, but agents also constitute a finite resource for us. I've numbered 11 locus for Prodry resistance and 14 locus for Downy resistance. It's a finite number. So we really need to, then this resource is a common good to everybody, and we have to preserve it. And the community, therefore, has to manage the resistance, has to design cropping systems that are durable in a non-selfish manner if we want to avoid the collapse of the system. Thank you very much.